All right, welcome everyone. Today is the second lecture on epigenomics, and we are covering joint learning across many cell types, epigenome imputation, and also the 3D epigenome. So on Tuesday, we talked about an introduction to epigenomics, the overview of epigenomics, antibodies, ChIP-seq, primary data processing, discovering characterization of chromatin states, and how to select the number of states and marks. Today, we're going to learn about how we can learn chromatin states jointly across many cell types, and about epigenome imputation, and then we're going to switch to the 3D epigenome lecture. So just to recap Tuesday's lecture, so there's one genome in every one of your cells, and it carries out every function in every cell type. How does it do that? How does it manage to achieve very, very different cell types and very different functions across every cell type? The answer is through the epigenome, through a collection of histone modification marks that function at the 200 base pair level for each nucleosome, a collection of CpG dinucleotide DNA methylation, which basically functions at the nucleotide level and says, you are repressed to regulatory regions that have the CpG methylation marks, and at the level of accessibility. So these are the three types of epigenomic features. And together, combinations of these features define different classes of functional elements. So we learn about the basic experimental technology for figuring out where are these modifications across the genome, namely chromatin immunoprecipitation, which allows you to build an antibody against a particular modification and then fish out all of the regions that have that modification and sequence whatever DNA comes with those regions. Everybody with me on this? We then map these sequencing reads that basically tell us what regions came down with it to the genome, and we get tracks like that. Everybody with me here? We also saw how combinations of many of these tracks can be basically used to infer these chromatin states. We saw how we can infer those tracks very rapidly by transforming our genome into this shuffled space that explicitly captures the redundancies of k-mers throughout the genome. And then we're able to search increasingly long suffixes of whatever query we have in order m time by sort of following these pointers and changing them every single time we include one more character because of this property of this Burrows-Wheeler transform. Everybody remember that? We then looked at various quality control metrics that allow us to sort of infer whether this is a good ChIP-seq experiment. For example, the strand cross-correlation of the left side and the right side that peaks exactly at the fragment length divided by 2. And we compared these read length versus the fragment length that sort of peak at these different distances. We also saw the art of peak calling, namely how do you take a continuous distribution and infer discrete intervals of high signal intensity and the diversity of solutions to that. And we saw how we can combine multiple data sets together by looking at the rank of each peak in one replicate versus another replicate, and then how far down the rank list does the reproducibility of a given data set fall off. And that allows us to choose irreproducibility discovery rate, or IDR cutoff. To learn these chromatin states from combinations of marks, we basically used a hidden Markov model, and specifically a multivariate hidden Markov model that has emissions, which are vectors of dozens of marks, and transitions that basically tell you how are these hidden states in relationship to each other on the genome. And then we saw how running this completely de novo learned model using expectation maximization onto the genome can basically tell us about different classes of promoter regions, different classes of transcribed regions, and how these in fact show a lot of biological enrichments. How the power is partitioned according to the information content, if you wish, of each of these states. Therefore, very few states can cover a big, big chunk of the genome that's kind of boring, and very many states are utilized to partition the most interesting parts of the genome. And then we were able to annotate 51 different chromatin states based on the specific enrichments of these de novo learned combinations of marks using independent data sets that were not utilized in learning these marks. And then lastly, we saw how with increasing numbers of states, we were able to capture increasingly the observed co-occurrence patterns of pairs of marks. If you only have 5 states, there's a lot of more co-occurrence in the observed data than what you would predict based on the product of the individual probabilities in your state. Whereas as you increase the number of states, this product of probabilities gives you the expected co-occurrence patterns, which are pretty close to what you actually have observed. Yeah. Absolutely, absolutely. So basically, one of the questions is, how do you know if you're overfitting? One way to do it is to partition the genome. To basically say, I'm going to use chromosomes 1 through 10 for learning the states, and 11 to 23 to learn the states again, and then ask how consistent are these states across each other. And we're going to get to that when we talk about different cell types in a minute, where we basically can relearn the states completely from scratch in every cell type and ask how consistently do I find the same set of states? And the answer is vary. And then we saw how the spatial information criterion was insufficient for selecting the number of states, but we could use this mixed approach, this nested initialization approach that will allow us to first learn a very large model that captures all of the interesting states, and then prune off the ones that were kind of redundant with each other, therefore keeping all the interesting things, but none of the redundant things that come with larger spaces. And that allowed us to basically see that once you have, using this nested initialization, recovered one of these states, you could then consistently recover it with independently learned large models. So that basically meant that, you know, for this particular state, this CNF state, for example, once I had discovered it here, I was consistently recovering it with any larger model because of this nested initialization. This takes away the randomness of this convergence away from the training. And that's, again, a technique that's very broad in any kind of machine learning model that you want to use. You may not have a high likelihood of capturing every low abundance state if you just train once, but this nested initialization effectively allows you to train as if you had learned a bunch of times, because you're capturing all these low abundance states and then prune away as you go. So that was sections 1 through 4 of lecture 8. So now we're going to cover section 5 and 6 of lecture 8 and all of lecture 9. So how do we learn these chromatin states jointly across multiple cell types? Up until now, we've only focused on one cell type at a time. And that's great because you can learn these combinations and you have this very simple HMM approach. But when you have hundreds of different cell types and dozens of marks across these cell types, the problem becomes harder. And in particular, you know, we'd like to learn a single set of chromatin states across, say, for example, 9 marks and 9 different cell types. So if you look at the data set, this is basically 81 different chromatin tracks. There are 2 to the 81 possible combinations of presence-absence between, you know, these 81 tracks. So how do you go about learning that? And specifically, how do you go about learning a consistent set of chromatin states so that you have the same set of emission probabilities, transition probabilities, and so on and so forth across the different states? And then this problem is important when you have 9 by 9, but increasingly we have bigger and bigger of these matrices. So for a paper that we're submitting in the next couple of days, we have 40 different chromatin marks across 834 different cell types. So the problem is much, much larger than before. So we're talking about 16,000 different combinations, that different individual tracks that we're trying to learn jointly. What are the different ways that we could do that? So do you have any suggestions for how I could go about learning these chromatin states jointly across these cell types if I have 81 different tracks? There's at least 3 approaches, so raise your hands if you have one of them. So I have this multivariate HMM that's very powerful and can learn across the whole genome. So I can apply it to these 81 different tracks in many different ways. What's one way? You seem to raise your hand as well. Yeah, go for it.
Right, because then you have your multi generate HTML, you just kind of dynamically program Burrows.
Yeah.
With kind of brute force its way through.
Okay, so how do you organize the data to give to your Baum-Welch algorithm? Yeah.
You combine the two subtypes together.
Yeah, yeah.
And make double the amount of states.
For example, go ahead. No, but I like your solution as well. I mean, they're both good solutions. So basically, one of them is combine them somehow, and the other one is maybe use different subsets of each data set. So combine them is the right answer, but there's multiple ways to combine them. So how, what's one way to combine them? Yeah. Yeah. So you could basically learn chromatin states in one of them, learn chromatate states in the other one, and then cluster the emission matrices and basically say, well, I learned this emission matrix here, slightly different emission matrix there. Let me match whatever state 1 I learned here with maybe state 7 that has the same emission vector. That's one way to do it. So that's solution number 3, I think. So just learn the states independently and then combine them, basically combine at the state level. Yeah.
Learn on them together and then afterwards split the split the subtypes.
Yeah, and then check if the frequency with which the marks occur in the state that I've learned match the emission probabilities of that state. In other words, if I learn jointly in both cell types, for example, I can then split up the cell types and then ask, is my model that I learned in cell type 1, for example, appropriate for cell type 2? So that's sort of similar to what you were saying about learning in each of them individually. So I can learn a set of chromatin states here, and then use that model there, and then say, if I were to run another iteration of Baum-Welch, would I get a different emission probability? I.e., are the frequencies, the counting frequencies with which each mark appears in the corresponding state annotations, matching the emission probability matrix for that state. And this is interesting because, I mean, you guys remember when we did this unsupervised learning, where you basically were, learning across the genome and letting that guide you and so on and so forth. Right now, you're basically saying, well, here's a new cell type that I've never seen before. Let's take the model untuned, apply to that cell type, and then see how much tuning would I need. Who's with me so far on this one? Awesome, great. And this is very helpful for you guys for any kind of data set that you end up with. You will very often have one data type or one data set and another data set that will be slightly different, and you'll wonder, well, how do I combine them? Okay. So in the context of chromatin states, one way of combining them is to basically stack them up. To basically, instead of having 9 different emissions that my multivariate HMM generates, a vector of 9, length 9, I instead could generate a vector of length 81, right? So I'm basically learning a joint model by pretending that I have 81 marks instead of having only 9 marks. Who can see this? Great. So that's one approach. That's the stacking approach. So the second approach, so basically that's the sort of the stacking approach. The second approach is to, instead of pretending I have 81 marks, to pretend I have only 9 marks, but across 9 length genomes. So instead of 23 chromosomes, that I have, something like 200 chromosomes. Okay? So you can basically concatenate the genomes of the different cell types and then learn chromatin states jointly. And we're already doing that in a way. We have 23 chromosomes. So we're concatenating all the chromosomes and then learning chromatin states on those. Might as well concatenate all of the chromosomes of all of the cell types. Okay? Who can see the difference between those two? Yeah? Awesome. So basically, in solution number 2, we go stack them. we learn every combination of activity as a separate state. And that basically means that I will have one state for embryonic stem cell specific enhancers that will basically say, aha, you know, this state corresponds to not only enhancers, but enhancers that are only active in that cell type. Okay, so then the number of states kind of explodes. Yeah? I kind of skipped it, so I'm going to go back to it. Yeah. But everybody with me on this one? So basically, the problem here is that my number of states explodes. With concatenation, well, that requires that all of the profiled marks were the same across all of the cell types. So in this approach, if I profiled 7 marks here and 20 marks there and 3 marks here, no problem, because I'm just stacking them up. Every mark is profiled, or at least every track is across the whole genome. So it doesn't matter if I have the same set of tracks. Whereas here, I better have exactly the same set of marks across all the cell types. Who can see the subtlety? Awesome. So that basically also ensures that there's common state definitions across all of the cell types. So basically, I'm learning the set of chromatin states once, and then the locations of where these chromatin states are can differ greatly from cell type to cell type. And then solution number 1 was simply learn the chromatin states independently in each of the cell types, and then after the fact, compare them, cluster them. So in this particular case, I'm showing you exactly that. So this is learning a 15-state model using 9 marks in each of 9 different cell types, and then asking, how often did I capture each of the chromatin states? So here, what I'm showing you is the emission vector, the states effectively, for each of those. And every state should be represented 9 times if it was learned independently in all 9 cell types, or say 11 times if in one cell type the same state was recovered slightly differently in 3 different ways, and in another cell type not at all, in another cell type two times. Is everybody with me? So this is basically allowing the model to say that it found different subsets of the states, and sometimes with different multiplicity in each of the cell types. And then you cluster them together and you ask, well, what are all of the states that I'm learning? And here you can see that, for example, this state here that has, you know, an emission of only CTCF is found pretty consistently across all of the cell types. And, you know, this one here is found maybe in a subset of cell types, and then there's subtle differences here and so on and so forth. Okay? Whereas the concatenation approach forces the model to learn a common set of states. And then you can basically ask, well, where are these states actually occurring in the different cell types? Okay? So the challenge of the concatenation is that if you have different sets of marks, like for example, this mark is missing in that cell type and that mark is missing in that cell type, you have to do something about that. One way is to skip this mark altogether. So, you know, this mark is missing in one of the cell types. I'm not going to be able to use it in the other 8. Sorry, I'm going to throw it away. And then this mark is missing one cell type, so I'm going to throw it away in all the other 8. So by the end of the 9 cell types, I'm left with very few marks that are common across all of them. Okay, so that's one solution. And it's fine. You can just tell the experimentalist, sorry, I'm not going to analyze your data until you give me a complete data set. Or you could basically use the probabilistic nature of hit Markov models to effectively treat the missing tracks as missing data. You can basically, in your expectation maximization framework, allow for unspecified data points. And as long as the pairwise correlation of the observed data is preserved, you might still be able to learn consistent chromatin states by integrating over all possible values of that data. Each possible value of that data weighted according to the remaining data that you have observed, and therefore sort of learn a joint model. In practice, this works quite poorly. And the reason is that there's just simply not enough information across these different marks to simply learn it using this linear model that this multivariate HMM assumes. But you can do something much more fancy, which we're going to talk about in the next section of this class, so in about 5 minutes, which is impute chromatin marks. So basically explicitly predict the maximum likelihood chromatin track for the missing data. So instead of integrating over all values of that missing data, just predict the maximum likelihood estimate of that data given all your other marks across cell types and within that cell type. And you know, this allows you to have peek calls for the missing data. This allows you to have all kinds of stuff for the missing data. Okay. Anyway, so we basically, you know, did that. So Jason Ernst and Brad Bernstein and several colleagues, we basically created exactly that. So a concatenation approach, not a stacking approach. And then we learn a set of chromatin states. And then we could basically say, well, how often are these covering each of the cell types? And how do they differ between embryonic stem cells and these immune cells, and so on and so forth, okay? And then we're able to annotate each of the cell types according to their enrichments, each of the states according to their enrichments in different annotations in matched cell types, okay? And that basically is very powerful because now, instead of having 9 tracks and 9 tracks and 9 tracks and 9 tracks in each of the 9 different cell types that you're trying to compare, you basically have a single annotation track for each cell type. And you can see at a glance what that tells you. That basically tells you that this region is active with an active promoter, active enhancers, active transcription marks in these 5 cell types. It appears to be inactive in the white ones and repressed in the gray one. And then it's in this funny state called a poised chromatin state, which basically shows both activating marks and repressive marks simultaneously. And that's pretty standard for embryonic stem cells, because embryonic stem cells can go down different differentiation paths, and therefore they maintain that pluripotency by having both active and repressed marks. It's like stepping on the gas pedal and the brake at the same time at the red light, and then you're ready to either go or stop, depending on sort of which way the differentiation landscape tells you you should go for that gene. Who's with me so far? Awesome. So basically, we're learning a single annotation track for every cell type. We're summarizing the cell type activity at a glance, and we can study activity patterns across the cell types. And that's where it becomes super interesting. We can now start building vectors of activity, and we can start correlating these vectors across cell types. So we can start learning that this is off, 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 on, and this is also off, 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 on, on. And therefore, if they're correlated, that might mean that this enhancer here is somehow controlling these genes there. Yeah. So in this particular project, we generated the data on purpose to be exactly matched across all of the cell types, and therefore we didn't have to throw away any of the tracks. But in new projects, what we're doing is systematic imputation. We're just completing these data sets. Was that your question? Yeah, yeah. So when we talk about imputation, I'm going to address exactly that, which is that for each mark, we can ask how much importance can I assign to that mark, completely unbiased, basically across all of biology. How much is that mark helping me learn? That's one aspect. The other aspect is, if I specifically care about poised enhancers, how important is each of the marks? So I can basically ask, how well can I recover each of the chromatin states with only a subset of the marks? So you can ask that question 2 ways, one unbiased and one state specific, and we're going to look at both. It's a great question. Yeah? In my view, stacking is pretty terrible because it explodes the number of states. And it doesn't allow you to say that this enhancer is now turning on. It just says, ooh, I'm entering this new state. And that state, what does it mean? Oh, that state is the state that corresponds to an enhancer active in that cell type. And that other state to an enhancer active in another cell type. So it's very difficult to actually make inferences about the, you know, the changes in activity across cell types. Whereas here, we know that there's a common alphabet, there's a common dictionary of states, and therefore we can see how we're switching from one state to the next. So it's a much more natural type of analysis. But you know, you can actually use the concatenation approach when you have dramatically different sets of marks. You have 2 marks here and 2 marks there and 2 marks here and 20 marks there. You know, it might be a fine approach. Any other questions? Yeah.
Just to clarify, So you have the same set of hidden states for all the cell types.
Exactly.
But are your transition and or emission matrices different by cell type, or are they the same?
No, no, you have exactly the same model parameters. Your emission and your transition parameters are the same. So it's as if you had a single very, very long genome.
Okay.
Yeah. Okay, any other questions? Great. Who's with me so far? Good, awesome. So now let's see what we can do with these activity patterns. So the first thing that I can do is basically, of course, expand this to many more. So we now have 127 epigenomes that we've run this in. And again, you can see correlated patterns of activity. For example, this gene here turns on in only those cell types. And you can see that this regulatory region here, again, turns on in exactly the same set of cell types, here and here and here, here. So I can start linking things together. So you can sort of infer information about the chromatin states. You can learn about how these chromatin states map to different regions of the genome. So basically, the first thing you can notice is that if you look at DNA methylation, some states are very highly predictive of what will be the underlying DNA methylation of that region of the genome, or the DNA accessibility. So some states are much more accessible, while other states are much less accessible, and so on and so forth. And the two tend to be inversely correlated with each other. You can also start painting the whole genome, and you can basically ask, what are the relationships between, say, gene density and the frequency of different chromatin states? And what you can see is that some regions of the genome are much denser in genes than others. Some are much sparser. And that actually corresponds to the set of states that are being utilized. So if you see these gene-sparse regions, they don't have any of these active chromatin states. They're mostly in this quiescent or in this heterochromatic state. Whereas if you look at these gene-dense regions, they fall into two classes. Some have these bivalent marks very pronounced, and others have these active marks very pronounced. And you can also see the chromosomal bands corresponding to this. So when you actually visually look at the chromosome, the regions that are darker correspond to these very gene-poor regions, and the regions that are brighter correspond to these very gene-rich regions. So there's underlying physical properties of DNA that are, in fact, one-to-one mapped to the chromatin state composition of different regions of the genome. The other thing you will notice is dramatic differences in their association with lamina. So in the second half of this lecture, we're going to talk about lamina association. These are the regions at the periphery of the nucleus where you usually put repressed segments of the chromatin. So these regions are much more associated with these very empty chromatin states, whereas these much richer chromatin states tend to be completely away from the lamina, as you can see here. Who's with me so far? Yeah. Okay, you can also learn about the relationships between these different cell types. For example, if you use one of these marks, this enhancer associated mark, to basically ask, how are these samples related to each other? You will basically see that all these blood samples are in fact clustering together. All these embryonic stem cell and induced pluripotent stem cells are clustering together. All these brain samples are clustering together, and so on and so forth. So that basically tells you that you can infer the common biology of all these marks by clustering their chromatin states. And you can basically see in a MDS, multidimensionality scaling approach, you can basically see that the dimensions that sort of separate this multidimensional space of that tree that I showed you in the previous slide are in fact allowing you to say that perhaps the most diverse cells, the first dimension of separation of all these different samples, separates immune cells from pluripotent stem cells. And then the second dimension, or the fourth dimension here, separates brain from muscle, and so on and so forth. That might explain a few things about, sorry. So basically from that, you can kind of see sort of what sets apart the epigenomic landscape of different tissues and cell types, okay? So let's now go back to these activity profiles that I showed you and see how powerful they are, okay? The first thing that they allow you to do is start linking enhancers to their target genes. So when you basically see this correlation that I mentioned earlier between these regions and those regions, that allows you to sort of establish links between these regions. And you can do that systematically by clustering enhancer regions based on these activity profiles. So you can build a vector of activity across these 127 cell types for every enhancer. So you have 2.3 million enhancer regions. You can basically build a 127 long vector for each of them, and then cluster these vectors and see what are the clusters that emerge. And the first thing that you'll notice is that most of the clusters are extremely tissue specific. So you can see here that, all of these clusters, for example, all of these clusters here are active in T cells and B cells, not in the other cell types, which is remarkable. And only about 10% of these clusters are in fact active quite broadly across many different regions. You get exactly the opposite picture if you look at promoters rather than enhancers. So enhancers are much more cell type specific, whereas promoters are much more, ubiquitously active across cell types. And then you can basically ask, for all of those states that I've learned, for all of those clusters that I've learned, what are the corresponding gene functions? So you can basically look at the genes nearby and look at their enrichment in common gene ontology terms. Okay, so we've talked about in the clustering lecture how you can evaluate the clustering by looking at an independent data set that was not utilized. In this particular case, we carry out the clustering using just these activity vectors, and then we ask, are the genes nearby, something completely independent, enriched in common gene ontology terms? Who's with me so far? So you're basically taking all of the locations that have that pattern of activity, and then you're asking, are the genes nearby, when I bring them together from the whole genome, are they in fact coherent in their gene functions? And that's what leads me to find that in fact these T cell and B cell enhancers are very active in immune gene ontology processes in pathways. And all of these embryonic stem cell active enhancers are very involved in genes that are active in development and morphology. All of these enhancers associated with brain are near genes that play roles in learning. All of these enhancers active in muscle are near genes that, play muscle-related roles. Yeah.
When you are saying that near that, are you talking about for activity space or like an extra?
Yeah. So such a great question. So basically, the question is, how do you find what genes are near these enhancers? So I have an enhancer here. How do I find the nearest gene? One way to do it is to look genomically at what are the genes nearby. Just basically, if you walk out in genomic coordinates and you find the nearest gene. A much more sophisticated way is to basically say, well, what nearby gene also has a correlated pattern of activity. In this particular case, we just looked at the nearby gene to be unbiased, but there are ways to do it with looking at pattern of activity, which can be very powerful. Any other questions? Okay, so basically this was looking at the correlation between enhancer activity and gene expression. So can we use the two to basically link things together? And using that, you can basically say, well, I have an enhancer here. What is the most likely target gene? And you can basically look at the vector of gene expression levels for those two genes, and then assign your enhancer to the gene that has, higher expression. And when you do that, you find that all kinds of things get better in terms of enrichments, in terms of functional categories, co-expression patterns, and so on and so forth. So that's basically looking at enhancer to target gene, just this part. But you can also use other vectors of activity. You can basically say, let me look at the enhancer activity pattern, and then ask if I also have correlated expression of a regulator. So now I'm trying to find who controls these enhancers. And you can do that by basically saying, well, which regulators show enriched motifs in those enhancers and expression patterns that are positively for an activator or negatively for a repressor correlated with my enhancer chromatin state. So now I'm going to basically use those three vectors of chromatin state activity, so basically whether the enhancer turns on or off, whether the upstream regulator is plus or minus, and whether the motif is enriched or depleted. And using the three together, I'm going to predict both activators and repressors, and also link the regulators to their target enhancers. Okay? Who's with me so far? Raise your hand if that makes sense. Awesome. Great. So the next picture is going to be a little complicated because I'm going to be showing three-way correlations. So the upper left corner of every entry, I'm going to be showing you the motif enrichment or depletion. And on the bottom right corner, I'm going to be showing you the positive or negative correlation with the expression of the transcription factor. Okay? So it will seem complicated at first, but hopefully it'll become simpler soon. So I now have different clusters of activity. So I have cluster A, cluster B, cluster C, and so on and so forth. That basically tells me the vector of activity of that group of enhancers across the 9 cell types. Who's with me on this part? Awesome. I then ask, for that vector of activity, do I see an enriched or a depleted motif for the corresponding transcription factor? So if I look at the OCT4 transcription factor, I can ask, is the motif of that factor enriched in this set A? Who's with me on the upper left triangle here? If it's enriched, I paint it red, and if it's depleted, I paint it blue. Okay? And then on the bottom right triangle, I basically say, is the transcription factor expression correlated with this vector or anti-correlated with this vector? If it's correlated, it's green, it's yellow, and if it's anti-correlated, it's green. And if it's not correlated at all, it's white. Who's with me on the bottom right triangle? Awesome. So now, using this, I can basically predict whether each regulator is an activator or a repressor. And an activator basically has positive correlation and enriched motifs. A repressor has negative correlation and depleted motifs. Why depleted motifs? Because if it's a repressor, when the repressor is expressed at that cell type, then all the regions that contain the motif will be turned off. So therefore, the regions that have the motif will be depleted for that activity pattern. If the regulator is expressed, the corresponding regions are depleted. Okay? So we can basically do that to sort of predict, for example, that OX4 is a positive regulator of embryonic stem cells, which we know independently it is, and that GFI1 is a repressor of these immune cells, which we independently know that it is. And we can do that across many different clusters. So we can basically look at all of the clusters of coordinated activity and then predict who are the activators and who are the repressors of those clusters. Raise your hands if you're with me. Awesome, great. Who thinks this is kind of cool? So we're basically using all these correlations to basically infer basic biological principles and how they teach us about the circuitry of the genome. Everybody with me? Any questions? And of course, we can extend that to now 127 different cell types, 200 different clusters, and dozens of different motifs that are either enriched or depleted in each of the clusters, and then using the expression correlation, infer whether the corresponding regulator is an activator in orange or a repressor in purple. Everybody with me so far? And from that, we can basically infer a circuitry of who are the regulators and their corresponding motifs that control enhancers active in different clusters of tissues or cell types. Great. So all of that is using this concatenation approach to basically learn chromatin states in a very small set, like 9 cell types, in a very large number of marks, 9 marks. But the problem is that as you start expanding the number of marks, you basically have a very sparse matrix. So in this particular case, the observed marks are in purple, and then the missing marks are in red. So we have missing data. So we would love to learn more complete models, but we can't because we simply don't have the data. So what can we do instead? Instead, what you'd like to do is imputation. Imputation basically means you're using a partially completed data set to complete the data set using correlations. So the problem is that we're trying to predict a mark in a cell type specific way across the whole genome, assuming that there's absolutely no data for the data set you're trying to predict. If there was some data, if you had, say, a chip-chip experiment on a subset of the genome, that's trivial almost. You basically just learn a machine learning predictor for what will that mark look like, considering that I've seen that mark in only one chromosome, can I predict the other chromosomes? But the problem is that technology doesn't work that way. For us, it's the same effort to profile one chromosome or to profile the whole genome. So if you do the ChIP-seq experiment, might as well sequence whatever you get out, and then you have predictions across the whole genome. So it's not very helpful to do this type of supervised learning in that way. Instead, we would like to assume that there's absolutely no data for the data set that we're trying to impute. So I've never actually observed this regulator or that mark in that cell type. How can I predict it? And the idea is that we're going to complete this very big matrix between, many marks and many tissues by combining potentially hundreds of data sets together in that prediction to generate data that will sometimes be more robust and higher quality than the observed data, okay? Here's why this can work. So if you look at H3K4ME1, this enhancer associated mark, the true answer is that in gray here. And then there's all these other marks that are actually kind of highly correlated with that right answer. But of course, you can't know that in advance because what we're trying to do is predict it without seeing it. And just to give you a preview, the red is how well we can predict it without ever seeing it. And you can see that the red and the gray are pretty identical. Similarly, if you look at H3K36 trimethylation, this is a transcription-associated mark, you can now see that, again, the mark that we're trying to predict is quite similar to a lot of other H3K36 trimethylation datasets, transcription-associated epigenomic datasets across many other cell types. So that means that there should be enough information either across cell types for the same mark or across marks for the same cell type to help with our imputation. So the strategy that we used is the following. You basically have a target mark that you're not allowed to observe. So you don't know what is correlated with that mark because you're not allowed to observe it. You don't have it. But instead, what you can say is, well, I don't have that mark in that cell type, but I can look at that mark that I'm trying to predict in other cell types and basically say, in this cell type, that mark, in this cell type, what is it most correlated with? And in that cell type, what is it most correlated with? And in that cell type, what other marks is it most correlated with? And therefore, you can learn a predictor in each of these other cell types for that mark that you're trying to predict. Who's with me on the left side here? Raise your hands. Awesome. So I'm basically going to learn a set of predictors using whenever that mark is observed in some other cell type, I will learn a function that allows me to predict that mark from that other cell type. Okay? And I can have lots of missing data because in each cell type, I'm learning a different function that basically uses only the data that's available in that cell type. And similarly, I can look, I don't have that mark in that cell type, but I have that other mark in that cell type, and I have that other mark in that cell type, and that other mark in that cell type. So I can learn what are the cell types that are the most informative for this cell type across other marks. So I'm basically using both the cell type to cell type similarity as inferred from other marks and the mark to mark similarity as inferred from other cell types. Raise your hand if you're with me. Awesome. So combining the two, I can now try to predict that missing mark in that cell type. Sounds good? So for this, we basically use the random forest approach. We use a combination of, it's actually a regression approach embedded within this random forest. And then what that resulted in is what you see here. And I'm going to zoom in the same region. So you can see here the observed marks in blue and the imputed tracks in red. So I hope you can notice that at extremely high resolution, these are individual 25 base pair windows, we can capture the extremely high resolution positioning of each of these marks. And as I zoom out, we can capture the large scale activity patterns of each mark across many different cell types. So here I'm showing you a random set of epigenomes, capturing all of the different marks that I'm trying to impute. You can see here at increasing resolution, so I can zoom in here and I get that, and I can zoom in here and I get that. And that basically means that I can capture observed data extremely well. Okay? Who gets it? Great. Any questions? Yeah.
Why are you doing this? Why are you trying to infer such a data?
If you're trying to learn chromatin states across 100 cell types and all of the marks are not available, you have to do something. So basically what we're trying to do is leverage the correlation patterns across all of these different marks to complete the data set. So if I'm missing, if I have one bad data set, there's nothing I can do. I have to throw away all of the cell type for that mark. But if I can impute it, I can basically, or I have to throw all of the marks for that cell type. So basically, in order to have a complete matrix, I can actually impute these data. Does that make sense?
But we're not adding any more information.
Well, you'd be surprised. You're adding a lot of information. The reason for that is that you're basically, as these data sets are getting more and more complete, you have a pretty good idea for what this mark will look like, even though you've never observed it. And it's not just, oh, I'm going to find the closest epigenome, because at every location of the epigenome, you're using a different prediction. So you're basically actually generating new information because you're capturing a lot of non-linearities in this sort of random forest regression approach. Yeah?
So do you do new imputations also correlating capacity?
Oh, that's interesting. So again, the observed data was never utilized in the prediction. Basically, we're building a predictor without ever looking at the observed data.
Sorry, not the observed data, but once you impute a track.
Yeah, I've imputed this red track here. Yeah.
Do you use that in future?
Oh, I see what you mean. Yeah. It's a great question. So basically, one way to do it would be to only use observed data and only predict based on observed data. Another way to do it would be to do a first round where I impute everything and then impute each of them using all the observed data, and then use the imputed data to do a second round of imputation where I now have much more complete data set. We only did the former. The latter is a great idea. We didn't do that. We basically only used observed data in predicting missing data. That is true. Yeah. Because right now, after you've predicted this track, you're now using that information. But you would not be able to use that to say, oh, look at how well I'm capturing the observed data. That's the only thing you would not be able to do. But it might make an overall better imputation because you don't care about circularity if in the end you're not going to say, oh, and look, it matches the observed data set. If in the end you're just trying to make a great data set, that's a great approach. Yeah.
Practically speaking, how much of your data can you impute before the model you're building out of it is not?
In this particular case, we imputed something like 85% of the data. So all the red stuff is only imputed, and about 15% is only observed, is also observed.
So a lot.
So a lot. And for these gaps here, where you have pretty complete data, it works very well. For these ones out there, it doesn't work as well. So basically there's some threshold. And I'm going to show you some graphs that basically tell you how well can I impute based on what marks are present. Yeah.
Maybe I'm misunderstanding.
Yeah.
It seems unlikely that you should have to impute so much of your data, right? Like, shouldn't you when you expect to observe a lot of marks in your cell types if your imputation shows that they should be there?
Yeah. So let me clarify what we're doing, and that's a great question, by the way. The reason why data is missing is because nobody did the experiment. basically there's a giant consortium that went out and profiled 4,000 data sets, but that matrix has another, 16,000 missing data sets. So basically what we're doing is not that, oh, we searched for that mark, but we couldn't find it. No, nobody did that experiment. Basically, it's a ChIP-seq experiment, it's a chromatin imputation experiment that was never carried out. It's not a mass spec experiment, for example, where I'm trying to pull down regions and then some regions are just simply missing. I'm like, oh, well, they must be there because I have these other ones. Does that answer your question?
Yeah, is it just impractical to do ChIP-seq for every single one? For every single
It's extremely expensive. Yeah, that's part of the reason why we're doing this. Yeah. Yeah. So this one and that one and that one, basically. Yeah. So for these, we had these 3 observed data sets. These are the blue ones. Here we only had observed data, but we did not impute anything for that column and for that entry. And the reason for that is because I didn't have anything else for that cell type that I could base it on. But yeah, using a single mark, we were able to impute all of these red guys for that cell type. But basically, whenever you see purple, that means that it's both observed and imputed. Whenever you see red, it means this is only imputed, but we had something that we could impute it from. And when you see blue, that means that I didn't impute anything there because I didn't have any driver that could tell me here's what it's similar to. Yeah. A little louder. That's such a great question. The answer is basically yes, but we don't bother. But yes, if I had 3 more data sets here in the middle, I would have to relearn the whole model and do a better job at imputing. Or, I mean, in practice, this would only affect, no, it would affect everything. Basically, the question is, do you only have to do that cross when you have a new data set? And the answer is that no, because that new data set teaches you about relationships between these marks and between these cell types that can now be helpful everywhere else. So what we usually do is that we freeze the data, we learn, and then we impute everything. And then 5 years later, when a bunch of holes have been filled in, then we can do it again. Pretty much, yeah. But for the chromatin state, it's a little different because what we are doing nowadays is that we have these chromatin state parameters that we've learned, and then we just apply that model to a new data set. We don't relearn each time because yeah. So basically, I feel that the chromatin state definitions are pretty stable after you've looked at 50 or 60 cell types, and you don't have to relearn those every time. So basically, the model that we learned in 2015 in the 127 epigenomes, we now reuse in 2019 in 1,000 epigenomes, because we feel that it will be just noise on sort of how slightly different they might be. I don't feel that after 127 cell types, there's some chromatin state that we've just never observed. Yeah, so the number of states, again, we had a whole section on that, on sort of how many chromatin states to infer. Right now we're sort of dealing with just 18 chromatin states, for example. We don't bother learning 50 each time because A, it's cumbersome, B, the pairwise exchanges across cell types start to become very sparse. Remember that sort of that curse of dimensionality that we talked about a few lectures ago? Like as the bins get smaller and the dimensions get bigger, you have very little data in any one spot. So we wouldn't have enough to sort of say, how is enhancer version 18 changing to enhancer version 13? It would be very few such events. These are all great questions. There were some more hands? Okay. All right. So then the question is, how well are we doing? The first thing is we can compare to observed data, and we match observed data, which is great. The second is that we can look at not just one location at different resolutions, but thousands of locations across the genome. So we randomly sampled thousands of columns, and we asked, what does the observed data look like across thousands of columns across 127 genomes versus the imputed data? And you can see here that the patterns match quite accurately. So that basically means that even across these thousands of random locations, I'm able to sort of recapture the activity patterns systematically. Is everybody with me here? Then the next thing we asked is how well does that compare to, say, always predicting the signal average, or always choosing the single closest epigenome and using that throughout the whole genome. So basically, I'm gonna, when I'm looking to predict a new cell type, I'm gonna say, oh, well, don't just predict something new, just go find the one in your data set that you think is gonna be the closest and use that one, okay? And the answer is that we actually do a lot better than even finding the best case single epigenome. So basically, for nearly all of the marks, red is higher than either this best case single epigenome or the signal average, which is quite remarkable. But it basically means that it's not a linear problem. I can't just choose one and be done with it. I'm really using combinations which at different places in the genome are putting in different types of information. Yeah.
Do you like that graph that you did slide?
This one?
No,
this one?
this one.
Oh, yeah.
Chromatin is also like much more consistent across different cell types than the others. Why do you think that is?
It's because we sorted it that way. No, I'm actually serious. Yeah, we could have sorted by blue, and then blue would look very consistent, and red would be jumping all over. Yeah, sorry, it's just a visualization artifact. It's higher, but I wouldn't say that it's more consistent. Sometimes it's down here, sometimes it's up there, just like those guys. Yeah. Okay, so then we can ask, well, am I kind of always averaging out my signal, or am I actually capturing dynamics in my signal? So if you look at observed data across cell types and how pairs of epigenomes relate to each other, you can basically compare these 127 epigenomes versus 127 epigenomes and ask, am I seeing that immune cells are like other immune cells, but not like non-immune cells? And the answer is overwhelmingly yes. So these groups of cell types, even though I never told the ChromaImpute program about these groups, it basically found that it generates epigenome relationships that capture these group boundaries. In fact, much more so than the observed data. This is kind of mind-boggling. It basically says that even though I'm imputing based on observed data, the imputed data has better dynamics of relationships of those cell types. And this is true across many different marks. So not so much for promoter marks, which are kind of boring, as I mentioned earlier, but for K27 acetylation, which is this enhancer mark, active promoter mark, accessibility, heterochromatin, polycomb repression. All of these different marks are basically consistently capturing relationships between cell types much better than observed data. So at first we were comparing imputed data to observed data, saying, oh great, well it's much cheaper because we didn't have to do the experiment, but it's almost as good. Look, they kind of match. Now we're basically saying, whoa, imputed data is kind of outperforming observed data, especially for something that you would expect observed data to be better at, namely capturing the dynamics across different cell types. So that was kind of weird. And then we basically said, well, let's look at how well they capture gene annotations. So transcription start sites and protein coding annotations. And imputed data is dramatically better than observed data at capturing known annotations, both for promoter regions and for transcribed regions, which is again quite mind-boggling. And then we basically said, okay, well, great. When observed data disagrees with imputed data, which one's wrong? And what we found is that when they disagree, when they show the least agreement, these data sets are also bad data sets, according to our other quality control metrics. So that's kind of cool, right? You can actually predict the data set better than you can observe it. I mean, my experimentalist friends are basically telling me, should we stop doing experiments? What would be your answer? The answer is obviously not, because we want to have thousands of your experiments to make better versions of those experiments. because we're basing our prediction on the observed data. But every single time, what are we doing? We're basically capturing information across dozens of data sets. Any one data set might be noisy because we didn't sequence it deeply enough, because I sneezed, my hands were shaking, a different technician did it, the antibody didn't quite work out, and so on and so forth. But all of these slight noises that affect any one data set are eliminated when I combine many data sets, many informats, in predicting any one imputed data set. Okay? So we can actually use imputed data as a QC. Agreement with imputed data basically tells you, well, when you disagree, you've probably done the wrong experiment. In fact, in some cases, all of these quality metrics were great, but it was simply the wrong antibody. So it looked like a great data set, but it was the wrong data set, or it was the wrong tissue, or the wrong cell type, or something. So we could find mistakes where there was an antibody swap that would have never been picked out based on those traditional quality control metrics. Okay. And then I mentioned earlier that we can use this to now ask, which marks should I profile in order to capture all marks? Which marks are the most informative? And which marks can simply not be well imputed? So here I'm asking, how well can I impute each of these marks using different combinations of features? And, you know, for example, all of these marks can't be imputed very well using each of those. But using only a core set of 5 marks, I can impute them pretty well, and so on and so forth. So that allows me to now prioritize which data sets I should generate as a core in order to predict, you know, the maximum number of marks. And then we also saw that imputed data is actually much better at covering, at understanding genetic association studies, which we're going to talk about much more later in the term. So, you know, we compared how well is the observed data capturing genetic variants associated with different diseases versus the imputed data. And the imputed data was capturing many more associations with disease that, you know, make a lot of sense biologically. And then we're able to learn these chromatin states. We've now repeated this exercise across 833 tissues. That's the paper that we're trying to submit with Kellis very soon. We've basically now generated this data across 833 different cell types, across dozens of different marks. And we can learn all of these gene ontology enrichments. We can learn all of these regulatory motif enrichments. And we can learn all of these genetic annotation enrichments across hundreds of tissues and cell types. Okay? So that was the end of lecture 2, or sorry, lecture 8. And we're now going to get into the second half of today, which is lecture 9, which is the 3D epigenome. And it's okay because, you know, it's okay. You know, the epigenome is compacted by a factor of 10 to the 6, as I mentioned earlier. So we're going to compact this by a factor of 3, which is not as much worse. But what we learned about is overall epigenomics, how to primary process the data, how to discover and characterize chromatin states, how to choose the number of states, how to learn these chromatin states jointly across different cell types. We talked about stacking versus concatenation versus independent learning, how to define these activity profiles and use them to link enhancers with their upstream regulators and their downstream target genes, and how we can actually impute epigenomes by completing these matrices, sometimes better than the experimentalists can actually profile them. Who feels that they've learned stuff? Yay. All right. We're still going to do a 30-second stretch break. So please stretch, and then we're going to talk about the 3-dimensional genome. You're going to miss out. Again, if you want to see the 1.5-hour version of this lecture, it's online. So I can do whatever I want. I can completely mess up. All right, the 3-dimensional genome. So today, we're going to basically learn about methods for studying nuclear genome organization, how to process and normalize these data sets, how do we interpret these data, and what do they teach us about the organization of the nucleus, and how does that, how does that conserve across different species, And what does that teach us about the mechanism underlying all that? Okay. So this is what the mammalian nucleus looks like. You know, the cell is all the black stuff, and this is just the nucleus. And the nucleus within it has these nucleolus, but it also has, you know, a lot of nuclear pores that allow the nucleus to communicate with the cytoplasm outside it. It has a lot of transcription sites where, you know, the DNA is transcribed into RNA that will then exit the nucleus and, you know, produce protein in the cytoplasm and so on and so forth. Okay, so there's a lot of organization and structure. And when you visualize it, this is what you see. This is the nucleolus that I showed you earlier, this big thing. And there's a lot of DNA in black, which is stuck to the nucleolus, and also stuck to this outside periphery of the nucleus called the lamina. So the cytoplasm is outside, and the nucleus is inside, and the DNA is in black, and the nuclear lamina is this periphery. Everybody with me here? So this is just basic cartography. And this is 6 micrometers wide, when in fact every single cell has 2 meters worth of DNA. Okay, so we're talking about a factor of compaction of 10 to the 6. This is mind-boggling. To understand what we're looking at, I'm just going to sort of give you analogies at different scales. So basically, again, there's 2 meters worth of DNA packed inside and 10 micrometer wide nucleus. So that's 10 to the 6. And if you look at the thickness of that fiber, it is 2 meters long, but how wide is it? If you have a rod that's like 2 meters long and you make it only 1 centimeter wide, it starts bending, right? If you make it 1 millimeter, it's crazy. Like, ooh. This is 10 to the minus 9 thick, okay? So it's 1 billion times longer than it is wide, okay? That just, you know, it's very difficult to fathom. So let's think about a tennis ball. A tennis ball is, you know, if your nucleus was the size of a tennis ball, then you could fit effectively 1,000 kilometers worth of string inside. That string would only be one, this is a little messed up. So if you, yeah, no, actually, no, this is about right. Yeah, so 20 kilometers and, oh yeah, there you go. That's a kilometer, sorry, 10 to the 3 is a kilometer. So you're basically fitting 20 kilometers into one tennis ball, okay? And if you have one of these Giga inflatable things that's about the size of a human, okay, slightly bigger, then you can fit 1,000 kilometers inside that, okay? And the string would only be 1 millimeter wide, okay? So imagine a string which is 1 millimeter wide, and it goes from here all the way to Miami. And you're packing that string inside here, okay? So that's the problem we're facing. Everybody with me? So much more intuitive than, you know, thinking about micrometers, okay? So how is that possible? That's possible because of many, many different rounds of compaction. So you've all seen this picture in your biology textbooks. That happens very rarely. That's basically when the cell is about to divide, it packs up all the DNA in a straight line and then divides, and then the DNA goes back to being a blob, okay? So most of the time, the DNA is not looking like that. How is it looking? Well, at the, you know, very, very high resolution level, it's a double helix. And then that double helix is packed up around nucleosomes. We just learned that. And then these nucleosomes are organized into chromatin. And sometimes they're stacked and compacted in heterochromatic regions. And sometimes they're loose and open in enhancers and promoters in euchromatic regions, okay? But all of these layers of compaction have been a mystery for many, many years until very recently, until the methods that I told you about. If you stain every chromosome with a different stain, using a different combination of dyes, what you basically find is that chromosomes actually in that blob form different compartments, which is fascinating. It basically means that there's chromosomal territories. And then the question is, what's missing? And what's missing is a high resolution cartography of where is every segment of DNA inside the nucleus, okay? So let's look at how we can actually do that experimentally. The first thing is we can map what regions are in the periphery. How can we do that? We can basically create an assay that couples the protein that makes up lamina, so lamin, very creatively named, is this lamin B protein, for example, is found throughout this green region. So what we can do is create an assay that will trick lamin B to deposit a mark on the DNA whenever it contacts that DNA. And then we can go and capture all the regions that have that mark and sequence them and figure out what regions were associated with the lamin. Raise your hands if you're with me. Awesome. So that's what we do. Basically, we obtain DNA bound by this landmark and find those fragments. And we can use either ChIA-PET or DamID to do that, okay? And then you basically end up with all of the regions that were associated with the lamin, and you can see which regions were lamin associated above the line and which regions were not lamin associated below the line, okay? And that tells you about these LADs, lamin associated domains. Raise your hands if you're with me so far. Awesome. So these are the LADs in yellow, okay? Yeah.
It looks like like a pretty significant portion. Is
yeah.
How does that work if you only have like the inner circumference of the cell? Wouldn't most of the DNA need to be?
There's a lot of chunk inside the nucleus. If you go back to that first slide of the lecture, there's a lot of different stuff going on. There's all kinds of organelles inside it. There's all kinds of organization, transcriptional factors, and so forth. And what you usually do is that you pull down from these lamin-associated region little things that you want to transcribe, and you pull them from that periphery and you work with them there, and then they go back there. Does that answer your question? Yeah, there's a real estate problem basically. Yeah. Yeah, and if you look at this, it's not far from that. Like, look at this big chunk of black all around here, right? So yeah, that actually matches what you would expect. All right, so that's at the big scale. What about at the fine scale? So what you would like to know is interactions between pairs of loci, not just associated with the lamin, but also every locus in the genome. And there's a very cool technique for doing that, okay? And it's called Hi-C for chromatin conformation capture on steroids. So chromatin conformation capture was 3C, and then carbon copy chromatin conformation capture was basically 5C, and then Hi-C was like the ultimate thing. We can't get any more Cs. So what is this thing? So chromatin conformation capture basically means that I'm going to capture chromatin conformation. And how do I capture chromatin conformation? There's a very cool thing that you can do. You can basically imagine your spaghetti inside your bowl of ramen or something. And there's all these noodles like going around. And what you can do is use enzymes to cut noodles. You just randomly cut a bunch of noodles. And then uses another enzyme to just glue together cut pieces. Okay? And now if two pieces were proximal to each other and interacting, they're very likely to join each other. Okay? So then I don't care about the spaghetti that was sort of cut and rejoined here, but I care about the pairs of spaghetti that were sort of floating around and then cut and joined between them. Okay? So basically, I care about the junctions between one region of the chromosome and another region of the chromosome. Okay? How am I going to get those? I can basically put little biotin beads that only stick to the fragments that were cut. Okay? And pull down these beads and sequence across the ligated fraction. So one side will basically tell me, oh, I was in chromosome 9, and the other side will tell me, oh, I was in chromosome 13. And it'll tell me that 9 and 13 were kind of hanging out. Who's with me so far? Raise your hands if you're with me. Awesome, great. So that's the basic technology. So chopping up the DNA randomly and then religating it and then creating these chimeras, and every single time I find a chimera, I know that I ligated two pairs that were interacting. And I can enrich for these chimeras, and what I get out is this type of stuff. And this type of stuff basically tells me what regions interact and what regions do not interact. Okay? So there's another technology called ChIA-PET. So what ChIA-PET does, and again, cute little products out there. What ChIA-PET does is that it combines ChIP-seq, which basically pulls down with chromatin specific regions, and then it says who else was interacting, in that landscape. So it basically does this sort of cutting and religating, but in the context of a regulator. So you can now figure out for all enhancer regions, if I do a ChIA-PET with K27 acetylation, I'm pulling down all these enhancer regions and then asking who were they interacting with. Everybody with me so far? Good. So you guys have basically learned, what there is to know. So there's a lot of processing that has to happen in order to understand these high-C data sets. And what you end up with is these kinds of domains. You end up with these block-like structure that you can zoom into, and within these blocks, you can find smaller and smaller and smaller segments. So ultimately, what that tells you is that there's two types of chromatin. There's purple chromatin that interacts with other purple chromatin, and then blue chromatin that interacts with other blue chromatin. It's mind-boggling, right? It seems that there's two sides to the genome. There's like the west side and the east side, and they don't talk to each other, okay? And so you basically have the jets and the sharks, and the jets are only talking to other jets, and the sharks only to other sharks. So that basically means that there's two compartments of the DNA, and that, they're not talking to each other. And in fact, those two compartments match exactly the lamina-associated regions and the non-lamina-associated regions. So the lads and the interlads, which is a good analogy with the jets and the lads. Anyway, so the other thing to notice is that within this nested structure lie topologically associated domains. So basically, there's clusters that are bundled up together. And these clusters are basically known as TADs, or topologically associated domains. And then you can sort of learn about these TADs at higher and higher and higher resolution. And they teach you a lot about the organization of the chromatin at that landscape. Okay? So there's a lot of questions about sort of what creates TADs. And to cut to the chase, what we found is that, and you can watch the online lecture for all of that. What we found is that these topologically associated domain boundaries match lamina-associated domain boundaries, and that at the boundaries of these topologically associated domains, every single time I see one of those breaks, I basically have opposite facing CTCF motifs. That there's one protein, CTCF, that has a motif that's about 15 nucleotides long, and they're asymmetric. So what that led to is a model of loop extrusion. And that basically means that as CTCF binds here and here, facing in opposite directions, they're helping pull in through, chromatin regulators. They're helping to pull in these sections of DNA, basically effectively creating a loop, which is then the way that the genome gets folded in 3 dimensions. Raise your hands if you're with me so far. Awesome, great. So this loop extrusion model is now the leading model for how that immense 2 meters long DNA fits into something that's 1,000,000 times smaller, okay? So the other main thing to realize is sort of, these players are basically these complexes. They're these cohesin molecules are what sort of interact with CTCF to actually pull the genome through these loops. So they create these little rings, and then they pull the DNA through these rings, and that's how you end up with these domains, okay? So basically, the mental image that you should have in your head is these loops everywhere, which have now ended up in compacting the DNA. Everybody clear with that image? Great. So if you look now at these LAD domains across species, what you basically see that even though chromosomes have rearranged between human and mouse, if you look at this mouse chromosome, it matches 6 different human chromosomes, but the corresponding regions appear to have the same type of chromosomal organization, nuclear organization, that basically these regions that are orthologous between human and mouse are in fact both lamina associated. And these regions are both non-lamina associated. Who's with me on this one? Great. And then that basically begs the question of sort of what's driving all that, okay? And we basically carried out an analysis to figure out what's driving all that. We basically built this classifier using all k-mers between 1 and 8 to find the motifs that are driving this. And we assessed the performance, and we were like, wow, we get 90% accuracy. Let's look at the motifs. Let's find out what are the motifs. The motif length was 1. So we were a little disappointed. But basically, lamin-associated regions were AT-rich, and lamin-away regions were GC-rich. It's that simple. We had all these like machinery, and it turned out to be super, super simple. So you could actually predict this whole thing just based on, you know, whether you're AT-rich or not. And that is very similar to sort of what's driving these high C domains of sort of, you know, A domain and B domain, or A compartment and B compartment are basically separated away to a large part based on sort of just nucleotide composition, and to a much more high resolution part by the specific set of regulators that are driving this. So you shouldn't think that it's all AT-rich, GC-rich. It's simply that these large compartments are separated based on that. And within these compartments, there's a lot of additional control that happens by this loop extrusion and these CTCF binding sites that are binding there. Okay? So we learn about topologically associated domains. We learn about lamin-associated domains. We learn about DamID and ChIP-seq for mapping where the lamin is. We learn about chromatin conformation capture, about chopping up your noodles and religating them and sort of pulling down all the glue with biotin and then sequencing across the glue fragments to figure out which parts were interacting using these chimeras. And then how, when you carry out the analysis of these regions, you basically find these topologically associated domains whose boundaries very much agree with these lamin-associated domains, and that sort of have this plate pattern of purple only interacting with purple, blue only with blue, the jets with the jets, and the sharks with the sharks. Okay, and then that these are conserved across species, and that these are largely driven by very, very simple processes. One is the loop extrusion of the CTCF, you know, opposing things, pulling things through cohesion rings, and then creating these loops. And also this lamin association that happens largely through a GC-rich, AT-rich. Who feels that they've learned stuff? Who feels that they wanted more than 12 minutes? Maybe. All right, so for those of you who wanted more than 12 minutes, go watch the online lecture. For the rest, you got the essentials. All right, on Tuesday, I believe we have a holiday. Sorry, is it? It's next week, okay. So on Tuesday, we're gonna talk about regulatory motifs. The mentoring session will be at 2 p.m. tomorrow. So prepare your slides, prepare your presentations, and we'll talk at 2 p.m. tomorrow.